complex and it sounds like something from a sci-fi episode in which researchers can use a vector to modify, to snip, to edit genes to stop the sickle cells from making misshapen cells. And Dr. Malik is going to explain this in very succinct and simple details on how this works. And at the end of this forum, you all are going to have an opportunity to speak and ask questions for Makaya, ask questions for Dr. Malik. No question is too hard, no question is too easy. Ask what you want to ask. And if not, I have plenty, because this is something I am very passionate about. It's something that has interest to my clients, my patients, and I am really excited about the opportunity to offer more disease-modifying therapies as well to those living with sickle cell. All right, so one other thing I wanted to share was that Makaya, who's a 20-year-old young lady, shared with me. She said, if gene therapy did not help me, maybe it would help somebody else. And although there's uncertainties with gene therapy, she took a leap of faith to make sure that others have an opportunity to be cured of their sickle cell. So I'm really excited to start this forum today, and want to make sure we are okay to get started. And I have not heard back yet, but we will hold on and hang tight for a few more minutes until we get the Facebook Live going, and we will get started. Can you show the slide of the people, Claire? We're good? Okay. So we're going to go ahead and jump right on into it, because I really want to make sure we have enough time to ask questions at the end. And I also wanted to provide you with resources and places to go in order for you to get more information about gene therapy. So I want to introduce my first guest. Her name is Dr. Poonen Malik. Dr. Malik is the director of the Comprehensive Sickle Cell Program and the program leader for the Hematology and Gene Therapy Program at Cincinnati's Children's Hospital Medical Center. I want you all to give a virtual welcome to Dr. Malik.
Thank you.
And last, we have our sickle cell warrior, our star of the night. A young lady who is a trailblazer for sickle cell, who has put her life on the line to make sure that there are opportunities available for those coming behind her to be cured of her sickle cell. Her journey is just fantastic, and I'm so excited to hear more from Makaya this evening. Makaya is a 20-year-old sickle cell warrior and a young trailblazer who has participated in a gene therapy clinical trial. Along with working 2 part-time jobs, Makaya is a student at Columbus State University, and she's majoring in sign language interpretation. I want to welcome Makaya to our virtual forum this evening. Thank you for coming. A little bit about me. My name is Dr. Nina. I'm the founder of TOVA Community Health, and I have been named Small Woman-Owned Business of the Year in Delaware. I have developed a telehealth model to improve access to care for adults with sickle cell anemia, leveraging a partnership with Johns Hopkins University. I am also a senior research fellow at Delaware State University. Shout out to our HBCUs. I'm a senior research fellow at Delaware State University, and in my spare time, I enjoy the arts and music and playing the violin. Thank you all for coming this evening. So let's just jump right into it. And I want to start with you, Dr. Malik. What are the current treatment options for sickle cell patients? What are their benefits? What are their limitations?
That's, a very good starting question and a very loaded one. So, well, the current, I will just talk about the current disease modifying therapies for sickle cell disease. So you have hydroxyurea that increases the fetal hemoglobin and prevents sickling. Some patients have been put on chronic transfusions to prevent side effects. There is hematopoietic stem cell transplant, or commonly called bone marrow transplant, from a matched donor. And the FDA has recently approved two new drugs, voxelotor and adakveo. There's also another drug that was approved a couple years ago, I think, Indari. These are at early stages. Time will tell how good they are at really modifying sickle cell disease. But the first three I spoke about, hydroxyurea, chronic transfusions, and bone marrow transplant, are really the backbones of disease modifying therapies in sickle cell disease. And finally, after these new drugs, the things that are currently being in clinical trials is genetic therapies as potentially curative therapies for sickle cell disease.
So thank you so much, Dr. Malik. But can you talk about the limitations of some of these disease modifying therapies?
Yes, absolutely. So hydroxyurea is a wonderful drug. It has really changed the outcome of patients with sickle cell disease. It increases the fetal hemoglobin, that is an anti-sickling globin, and prevents the red blood cell from getting misshapen. However, it is a pill that has to be taken daily. And it requires medical monitoring of blood counts. And a few patients do not tolerate this drug very well. The most important limitation is really compliance of a daily lifelong drug, and the expense. In resource poor countries, it's actually unaffordable to take it for life. But when available and when medical care is available, proper monitoring, it's a wonderful drug. Chronic transfusions basically is like replacing the sickle red blood cells with normal red blood cells, and cause an immense relief in symptoms of sickle cell disease, are typically given to patients who've had a stroke or have had very severe disease. The downside of chronic transfusions is that you have to come in to get transfused. You need central lines, which come with their own set of complications that can occur. And they are associated with iron overload. So you need therapies to decrease the iron burden by giving all the transfusions that you do. So they do decrease the quality of life of a patient or increase the medical burden of sickle cell disease, but they provide very good relief of symptoms, as I said before. And the third is hematopoietic stem cell transplant, or bone marrow transplant. And this is given from usually a matched sibling donor, or a matched unrelated donor if they have registered in the national registries. And this is actually a definitive therapy that can cure sickle cell disease. The downside is that not everybody has a match. So only about 15 to 20% of patients, or 15 out of 100, will have this option, and the rest do not have this option. And then the other downside is that even though we try to match the tissues really well, there are still minor mismatches if it's an unrelated, especially if it's an unrelated donor. And that can cause immune side effects like graft rejection or graft versus host disease, and can require immune suppressive therapy. The newer drugs are too new to comment on the pros and cons. They've just been approved based on some early trial data, voxelotor, adakveo, and Indari, and time will tell. And gene therapies are currently being clinically tested.
Thank you so much, Dr. Malik, for that. But now I want to go to Makaya, and I want you to tell me about your experience growing up with sickle cell anemia.
Growing up with sickle cell, I wouldn't say it was easy, but I don't want to say it was like super hard either, because I feel like I adapted to everything that was thrown at me very well. So with growing up with sickle cell, of course, there were things that I had to learn how to do. I had to teach myself how to become ambidextrous, because if I had pain in my right hand, how am I going to write?
Oh, wow. I didn't think about that.
Yeah, so, and I'm dominantly with my right hand, I write. So I can write with my left hand if I need to. And there's just, like I said, there's different things that I had to teach myself how to do. And I had to teach myself how to, like, not necessarily get over the fact that my friends can go swim and I have to wait until everybody gets out at the birthday parties and things like that. But I most definitely made it work.
Did you miss a lot of school?
Yeah, I had to miss a lot of school for transfusions and like for crises and things of that nature. I tried to attend as much as I could. There would be times where I was in there meted up and dozing off and everything. A decent amount of school, yes.
And tell us about when you had crisis, where did you have the pain?
Majority of my pain came from my legs, majority of my right leg. Sometimes it would be in both. Or as I got older, once I hit my teen years, I started having a lot of back pain as well, lower back pain. And then sometimes, like I said, it would be in my arm.
And tell the viewing audience, what does a sickle cell pain crisis feel like?
Ooh. Personally, to me, there's different kinds of pain that you could feel. So the worst is like, you know how when they lay out pavement and there's still that big roller thing that has to like roll it out, it feels like somebody's driving one of those back and forth over the area that's hurting.
That's like a crushing pain. Wow. Wow. I can't imagine that, but that must be very, very painful. So Micaiah, tell us, what types of treatment have you tried?
I have done hydroxyurea. I tried Endari. I've done apheresis, and I've also done the blood transfusions.
Did any of those help manage your pain or help prevent complications or hospitalizations when you were having crisis?
The hydroxyurea worked. My mom was probably like, "Oh, why is she saying that?" Because it was a battle. I didn't really want to take it. And then the transfusions did, like apheresis worked well as well too.
But it also dried up your iron as well, and you had to come every month.
Yeah.
And you had to drive an hour and 15 minutes, right? One way and back to get the transfusion.
Yes.
That must have been a huge burden on your family.
Yes, with having to take off for work, my mom having to take off, or me having to take off for school. Yeah.
Wow. I thank your family for sacrificing so much, because you're such a lovely young lady. But I want to go and ask Dr. Malik to talk a little bit about what is sickle cell?
So sickle cell disease is a disease of the red blood cells. So the red blood cells contain a mutant or a abnormal hemoglobin called sickle hemoglobin. And what sickle hemoglobin does is whenever the red blood cells give up oxygen to the tissues, it clumps or what we call polymerizes into long fibers. And those change the shape of the normal round red blood cells to bizarre sickle shape or hook shapes. And these misshapen cells actually clog up the small blood vessels, causing immense pain, and they decrease the blood supply to the organs the blood vessels are supplying, and so cause tissue ischemia, pain, chronic organ damage that accumulates over time, and many other complications that are associated with the disease.
Right, yes, complications. So talk about, what is gene therapy? So how does it help to stop that hemolysis process or sickling of the blood cells so they can get more oxygenated blood to the tissues and the organs?
So gene therapy, as I said, sickle cell disease is a genetic disease. So gene therapy is actually either adding a normal gene or a gene that, replaces the function of the abnormal gene. So it corrects the function of the abnormal gene to a cell that makes red blood cells for life. And that blood cell is called the blood making cell or the bone marrow stem cell. So basically, in gene therapy, we do it in 4 steps, and as is shown in the slide. So first, we place the patient on apheresis or transfusions, as Makaya had just mentioned, and this is for 2 to 3 months. This is so that their bone marrow can become healthier, to be able to collect the blood making cells. Then just like you donate platelets, you go through apheresis. We do apheresis to collect the blood making cells from a vein, and this is done outpatient almost. So and then the third step is we take these blood making cells or the bone marrow stem cells to the laboratory, and then we mix them up with this super gene that here I have depicted a picture of the fetal hemoglobin gene shown with the Superman sign. We mix the cells with this gene, so the gene automatically enters the cell and sticks itself into the cell's DNA. And then the patient comes back and gets a dose of chemotherapy to make some space in the bone marrow. And then they get back their genetically modified blood making cells. So these are the 4 steps in which we do the gene therapy.
Thank you so much, Dr. Malik. Can you tell us how it is similar or different from a bone marrow transplant?
Great question. So for a bone marrow transplant, you need the blood stem cells from another person. The person has to match you. So most commonly, it is a sibling. Not all siblings will match, because 1 in 4 will be a match. And that 1 in 4 sibling in a family where there is a child with sickle cell disease you can have another sibling with sickle cell disease. So the probability goes down. And that's why I said about 15 to 20% of people have a match, not 25%. So you have to have a matched related or unrelated donor available for a bone marrow transplant. But for gene therapy, you are adding a gene into the patient's own blood making cells. So every patient is their own donor. Hence, it's universally available to them. The second advantage, if gene therapy is successful and becomes the standard of care, the second big advantage of gene therapy is that you're getting back your own cells. So there is no mismatch. There is no mismatch. There is no minor mismatch. So it does not require any immune suppressive therapy, and it does not have the consequences that you can have with bone marrow transplant. Some of the side effects like graft rejection or graft versus host disease. So those are the advantages of genetic therapies.
Wow. It seems like it's a game changer for sickle cell treatment.
Well, if it works, it can be a real game changer.
Thank you so much, Dr. Malik. So Makaya, I wanted to ask you, what problems or complications have you experienced living with sickle cell anemia?
The biggest complications that I've had were just really bad pain. And I also had acute chest a couple of times before. I had a blood clot before. But I haven't had like a stroke or anything like that, luckily. So it's been pretty minimal.
So acute chest is like having a bad pneumonia where you have whiteout in both of your lungs. Did you have to go to the ICU? Were you ever intubated?
I've been to the ICU before, but normally I was able to stay like on the hematology floor.
And I'm sure that was very scary if you can't breathe and you're struggling to get oxygen in your lungs.
Mm-hmm. Yeah.
Wow.
It's not.
I can imagine. Also, one of the things you share with me, Micaiah, is that you have been hospitalized about almost every month for the last couple years. Tell me how that's affect your quality of life.
Well, that was about like 2 years ago, a year and a half ago. It would only affect it when it was time for school, when I was still in high school, with having like tests coming up and like having to study and missing out and having to kind of teach myself what we learned in school, and going out with like friends and stuff like that. It interfered with that.
I could imagine that that must have been very challenging, missing a lot of school and trying to keep up with this schoolwork and all of that, being in the hospital and never knowing when you're going to get into a bad crisis.
Mm-hmm.
Did that cause you a lot of anxiety?
No, it never really caused me anxiety, just because I knew that like it was gonna happen, and I didn't want to. Because if it would have caused me anxiety, then I would have been stressing about it. Then it would have brought it on anyway. So there was no like
You have a
point
really
in
positive
worrying about it.
very
Wow.
positive attitude.
I wouldn't say that some others would be so positive when knowing that it's such a debilitating experience that you have to go through on a regular basis every month.
Mhm.
But so, how did you hear about gene therapy, knowing all of these complications that have you had, being in the hospital every month? How did you get intrigued by it?
I was asked when I was younger, around like 12, 13, if like if this became a thing, would I do it? Initially I was like, no, you know, I hadn't really experienced much life being 12, 13. I was just like, no, I wouldn't do it because this is all I've ever known. And such a drastic change would be really crazy. As I got older, started experiencing more things, and started seeing my friends with sickle cell pass away and things of that nature, it kind of changed something in me a little bit. So when they asked me, it was February of 2019, they gave me a call and was like, hey, you may be eligible for the gene therapy trial. Like, do you want to do it? And I was like, yeah, like, let's talk about it. So at the next visit after that, we talked about it and we met up with the team and everything, and we went with it.
Micaiah, tell us about your two friends who died from complications of sickle cell, because when we had talked, you said that was one of your catalyst sort of moments that made you want to get the information about this opportunity.
Yeah, so I had two friends. The first one, his name was Andre. He passed away in 2017. He was
How old was he?
20 years old when he passed.
Okay.
And then about a year and a half later, I had a 13-year-old friend that passed away from it. And I'm really close with her brother. I don't want y'all to be like, 13? Like, what was you doing hanging out with a 13-year-old? We're all like really close. We all went to camp together. So when she passed away, that one kind of hit me hard. And she passed away in February of 2019. So they called me like right after it happened. And I was like, let's do it, you know. I have a tattoo on my arm. It says, don't waste a moment. And then there's two birds that represent my two friends that passed away.
Oh, wow.
They never wasted a moment in life. I look at it, I say, hey, don't waste a moment. Take the opportunities that are given to you, because you never know what could happen. So.
Wow, to have to make that decision at such a young age and be so mature about it, I'm just so impressed. But Micaiah, I want to ask you, what concerns did you have about gene therapy in the beginning? I'm sure you had some people who were naysayers, some people were afraid for you. What is that? Why would you be altering your genes? And so did you get a lot of support? I mean, what concerns did you have?
The only concern that I had was like, it wouldn't work. I'm pretty like forward with everything. I'm like, let's do it. Where's the pen? Give it to me, you know? So I personally didn't have concerns. I personally still don't believe that it happened. So it still doesn't really seem real to me. But I'm sure that my mom and like friends had concerns, but my family and all of my friends were super supportive about it. And especially when it was time to do like the final step, everybody was like, do you need anything? Do you need us to switch out with you in the hospital and things of that nature? So everybody was all for it. Even random Insta followers were reaching out, and everybody was really supportive.
What kind of risk did they talk about when you had to sign that informed consent document? Did that make you scared at all?
Um, the risk was really like there was a long list of things. I'm gonna be honest with y'all. I'm not the best at sitting in meetings and reading like long lists of papers and things of that nature. So I kind of remember some of them. I kind of don't. But the biggest thing was it may not work. That was the biggest thing that stuck out to me was it may not work. So
Thank you so much, Micaiah, for that. So I want to go back to Dr. Malik to talk about what are the potential risks of gene therapy. You know, when Micaiah had to read that informed consent document, and you have to list those, all of the potential problems that could happen. And as Micaiah said, it may not work, but there's also potential benefits. Talk about that.
So as Micaiah said, the list is very long and we are required to inform our patients for each and every risk. So the list is very comprehensive and long, but I can hit the main points or the big things that are important and very specific to gene therapy. One of them is the risk of um, chemotherapy conditioning, which is given to make space. So um, and some gene therapy trials are using a smaller dose so that there's less side effects. So when you use a lesser dose, you make less space in the bone marrow for the cells you're giving back. And so there is a risk that it won't work, you know, but there is also, but if it works, it comes with much fewer side effects. The other approach a lot of people are trying, there are other trials that are trying, are they give 4 days of strong chemotherapy, which is the dose that you give for bone marrow transplant. And with that, you completely, kind of erase you basically kill off all the blood making cells that are in the bone marrow, you wipe it out, and then you give back you sow fresh seeds, which is the gene modified cells, you give them back. This is the same concept that is done in bone marrow transplant. So this approach has a higher chance of success, but it has a higher risk as well, and higher side effects, long-term effects of the high dose chemotherapy and the acute side effects. So that is one risk. The other major risk of gene therapy is we are sticking a gene or altering the genetic makeup of the cell by delivering a new gene or an anti-sickling gene or a normal gene. And that gene, you since it's a genetic therapy, you cannot tell. It could land at the wrong place. We can't often control that. So, and turn off or turn on a gene that's very important for the cell. And very rarely, with this type of approach that we are using right now in clinical trials, we haven't seen those adverse events. But in the older generation of gene therapy, about 10 years ago, this led to leukemias because the gene landed in the wrong place, and it was the genetic design of how it was carried in was such that it activated some cancer genes. So that was a side effect. After that, the field stopped and the entire field changed. The type of approach was changed. And I don't want to scare anybody because with this type of approach, there has been no such side effects reported, but there is always a remote possibility of that happening. And since that has happened, we inform our patients when we put them on any genetic therapy trial. Um, and those are the major risks, I would say, that are there with the gene therapy. There are many, many effects. Chemotherapy will temporarily lower your blood counts. So you may need some platelet transfusions, some blood transfusions, but they come back. The higher the dose of chemotherapy, the longer it takes for the counts to come back. The lower the dose, the quicker they come back. So with the reduced intensity chemotherapy, which is the kind that that you underwent, Makaya, usually it's about a week that the counts are low. But with the transplant dose of chemotherapy, or the larger dose, we call it the myeloablative dose, you can have low counts for about 25 to 30 days. And so when you have counts that low, then you can have some side effects. So these were the bad things about gene therapy. The good things is that the new cells that you give back that are genetically corrected are now going to produce red blood cells for the life of an individual. And these red blood cells will resist sickling. And that is the most important benefit of gene therapy, is it's a one-time treatment. So like hydroxyurea, you would have to take daily to increase the fetal hemoglobin. But with gene therapy, you can put in the fetal globin gene, and it would be a one-time treatment. And for the entire life, the blood-making cells would keep making red blood cells with fetal hemoglobin so that they don't sickle. So basically, that is the benefit of gene therapy, is a one-time approach using your own blood stem cells, and has the possibility of being given with minimal chemotherapy. And now, scientists are investigating ways of doing it even with non-chemotherapy agents, but that is still in very early research. But I think the future will be 10 or 15 years from now. We would just give them an antibody to make space and deliver the cell, so it won't have all the side effects that you see. But that isn't yet ready for prime time. It's still in the works in the laboratory.
Thank you so much, Dr. Malik. I also wanted to ask you, we're going to do a little bit of myth busting, because this is a question that a lot of people have shared with me. Some gene therapy trials use the virus in HIV. Can you get HIV from gene therapy?
No. The short answer is no. The long answer is that, scientists, in order to do gene therapy, you have to use a way of getting a gene into a cell. So if you think about it, every single day, even today, you must have eaten vegetables, meat, all of them, plant cells and animal cells, all contain a lot of DNA. Genes is basically DNA. And not a single DNA molecule gets into your body. Your intestines are bathed in foreign DNA, and yet it doesn't enter the cells. And that's because the body and nature has made every organism foolproof to prevent entry of foreign genes, right? So how do we get a gene into such an important cell like a blood making cell? And if you think about it, viruses have evolved over millions of years, and they have figured out how to enter a cell. So we don't actually ever use the HIV virus. So the virus has made various components. One of them is the smartness to enter the cell and stick itself there. And the second is to cause disease. So over time, we have figured out what is the recipe the virus uses to enter the cell. What are the sequences? And without ever using a virus, we just stick those sequences onto the therapeutic gene, like the fetal globin gene. So basically, we're never using an HIV virus. So you could never get HIV from this disease. You're just using a little piece of HIV, which is not going to cause any disease. It is just that information of how to enter a cell, and stick itself into the cell. And so therefore, I think that it's important for people to know that these, gene therapy is actually a mixture of several, we take information not only from HIV, but various other viruses, and and this whole apparatus is called like um, like it's almost like if you wear a cloak, and you hide the gene in that coat. So that coat is the recipe that kind of the cell fools the cell into believing that this is not a gene, and it lets it enter the cell. And then what you're delivering is not a disease causing gene. You're delivering a gene that actually is a therapeutic gene. It would do correction of the genetic defect.
Thank you so much. It's almost like the virus is stripped of its infectious component, and then you use that vector to carry that information into the host cell.
Absolutely. And it's also important. That's a very good analogy that you have given. It's basically, taking just the coating, but we never use the virus. That's another important thing, is we just know the sequences. So people in my laboratory work with these vectors all the time, and they work like any other chemical they work with, only because it's completely safe. We are not ever using any live virus ever. We are just taking the sequence that allows it to enter. But it is sequence derived from the HIV virus, so it's scary initially if you don't know how we are using it. But we cannot cause any disease with this approach.
Thank you so much for clarifying that, because I know a lot of people in the audience today have that concern and that question. So I'm glad that has been cleared. But I want to ask you, so how many patients have been treated with this type of lentivirus-based gene therapy, your approach that you have patented and invented?
So the gene therapy approach that we have invented was that I had worked on involved using a modified version of the fetal globin gene. It is a much more potent version. And we use the lentiviral vector to deliver it, and we give a reduced intensity chemotherapy. It's just a single dose, which we have figured will help and yet do minimal harm. So because our goal is not to do harm, we want to do this to cure and help patients with sickle cell disease and not do any harm. And so we developed this approach, and I think we have now treated 4 patients with gene therapy, including our star here. And then there are other groups. There are other groups that are also adopting similar lentiviral approaches to increase fetal hemoglobin using a different approach, or to modify the beta globin gene so that it doesn't allow the sickle hemoglobin to sickle. So there are various ways, there are different genetic approaches, but the bottom line is they all prevent the red blood cell from becoming misshapen. Then other groups are just trying, CRISPR Therapeutics is trying gene editing, where they basically go in and do an approach that will that's kind of a hit and run approach that turns on the fetal hemoglobin gene forever, a sleeping gene, it turns it on. So there are many approaches. The bottom line, as I said, is to prevent sickling, and that is the goal. I think that for sickle cell disease, there must be over, by now, I think about maybe 30 to 40 patients treated. For other thalassemia, which is a sister disease where with sickle cell, you make abnormal hemoglobin. With thalassemia, you don't make hemoglobin. And in that, probably about 50 to 75 patients have been treated. And overall, with lentiviral vectors, over 200 patients with other diseases, other blood disorders have been treated with lentiviral vector gene therapy. And the knowledge of these trials is up to 10 to 12 years they've been treated for, and it's stable. The success is there, and it's long-term, and we haven't seen any major side effects of this therapy.
Wow, that's fantastic. Can you share with us, are there other approved gene therapy approaches for other chronic conditions?
So the only, I think there are two gene therapy approaches that have actually become a standard of care now. One of them is for an immune deficiency called ADA deficiency. And this gene therapy approach was developed in Italy, and now it's licensed as standard of care because it has effectively treated so many patients and they've been corrected. Their disease has been corrected for over 10 years. And the second disease is a type of congenital blindness. And they have done gene therapy for that. And that also has been licensed as a standard of care now. So
Wow.
they inject the gene into the eye that's missing, and it goes and, restores the blindness. So in people, so those two are approved by the FDA. So there are many others. So gene therapy for thalassemia has been approved in Europe using lentiviral vectors. So that's a third one actually that has recently been approved. So there, as there is success, the FDA then gives approval. But I just wanted to say that gene therapy, because you had asked about, dispelling some myths, it has very strict regulatory oversight. So there are many, many regulatory bodies that oversee anybody doing gene therapy trials. We have the Institutional Human Rights Committee, or IRB. We have the Institutional Biosafety Committee that makes sure that the vectors we use, as you said, the myth is this HIV, they make sure that we are using the safest possible approach that will cause no harm. So we have the FDA, and we have the Data Safety Monitoring Board that continuously monitors all the data that's coming out. So, and we have to regularly report. It takes years to get approval, and then we have to regularly report all the findings. And if there is any side effect, the trial can be put on hold immediately. So it is done in the safest possible manner.
Thank you so much. So now I have a polling question, okay? So can we put up the polling question for our viewing audience, please? Okay, so everyone, I want you to submit an answer now. I hope you're been paying attention. Gene therapy holds the promise to cure the following conditions: sickle cell disease, beta thalassemia, Fanconi anemia, or all of the above. All right, guys, put your answers in, submit them, please. Couple more seconds. Let's see what we come up with. Do we have the results? Come on. Wow, 75% chose D. D is correct. These are all blood disorders that there's a mutation or a defect or a missing gene that causes the blood cells to make abnormal cells. All of these therapies can show promise in being cured. Anything else you want to say about that, Dr. Malik?
Yes, all of these are genetic. Sickle cell disease, as I said, is you have a mutation in the beta globin gene. Thalassemia, there are many beta globin gene mutations that can cause thalassemia, but you just don't make beta globin or hemoglobin. And Fanconi anemia is a defect that is a DNA repair defect, so the cells are unable to make enough blood-making cells. And this is also caused by defective genes that are well known. So all these genes can be added back to correct the disease with gene
That's
therapy.
exciting. That is so exciting. Now for the second poll question. All right. All right, here we go. The lentiviral vector inserts DNA or a functional hemoglobin gene into the study participants' own stem cells. A, true, or B, false? Okay, let's see what you guys have come up with. 100%. Love it, love it, love it. They've been paying attention, Dr. Malik. Very good. So as Dr. Malik says, that the lentiviral vector strips the virus of all its infectious components, and it's like the delivery vehicle. It packages up the gene and it transfers it into the host cell. Dr. Malik, do you want to give a more scientific version
No,
of that
you said it
response?
perfectly. You couldn't have said it better. It's basically attaching the recipe that lets the fetal globin gene enter and stick itself into the cell's DNA.
Well, I'm excited because our viewing audience is like getting these questions right. They're paying attention and they're learning. But I wanted to go back again to Micaiah and all of these risks that had to be disclosed to you. Were you concerned about anything in particular with enrolling in a gene therapy clinical trial?
The only thing I was really concerned about was the chemo, because I had never had that before, had never had to like have any sorts of interactions with it. I wasn't concerned about like losing my hair or anything like that. I had actually cut my hair like 6 months prior to getting chemo. And then I actually had them shave me bald before I went in so that I wouldn't have to watch it fall out. But the only concern that I really had was the experience with chemo.
And what intrigued you most about gene therapy?
It was the fact that I could live pain-free for the rest of my life. To be able to learn how to swim. That was my number one thing. I said, when this is over,
Good.
swim. Like, so.
Awesome. So all of those things that you didn't get to do as a child, you want to make up for that now.
I can't wait till it snows. I don't know what I'm gonna
You
do.
wanted to build a snowman, right?
Somebody gonna walk past, like, is she okay? Like, I am fine. Just know. Like,
and my last question is, who did you talk to weigh these very heavy decisions about the risk and the benefits? Who was your support team?
the research team at Cincinnati Children's, it was two ladies, Amy and Adrian, that those were our contact points. And then Dr. Grimley, who was like the transplant doctor, he was a part of that team as well. So if we had any questions or anything like that, we could reach out to them. But also my hematologist and nurse practitioner, they know some things as well. So I was able to reach out to them too.
Thank you, Doc. Thank you, Micaiah. So Dr. Malik, I wanted to just share, because there are some people in our community that are very concerned about this approach that is just transformative because of the history in this country and the history of ethical violations in clinical trials, especially in the events of misinformed Black American sharecroppers in the Tuskegee clinical trial that were had a diagnosis of syphilis, but then were denied treatment to try to understand the long-term effects of syphilis and how it manifests itself in many different stages. But how are trials conducted today? So how can we be sure that these abuses won't happen again in future clinical trials?
Yes, it is really unfortunate that all of these things happened in the past, but we have learned from those, and these days the human rights committees are extremely strict about making sure that a patient's rights are protected. So, as I said before, there are there were 5 regulatory agencies when we initially got approved for the trial. Now, one of the agencies, one is run by the NIH called RAC, one is the FDA, the Institutional Review Board or the Human Rights Committee at the institution, the Institutional Biosafety Committee, and an independent data and safety monitoring board that cannot have any person from the institutions that are conducting the trial. So they independently review the data, and they ensure, and we have to report all the side effects, everything, informed consent, how they review the entire informed consent and make sure that every information is being provided to the patient. And so there is basically the gene therapy trials are under a big magnifying glass, and they are being watched very, very carefully. And Makaya can probably tell you that she was given like probably a book in the form of an informed consent. Because it is, we are required to inform our IRB of any gene therapy side effect that arises in any trial. We are supposed to inform the patients. We're supposed to revise the informed consent. If, say, in a gene therapy trial in Fanconi anemia side effect, a major side effect, or a leukemia, or something big happened, we will immediately inform our IRB and inform the patient who has been in the trial that this has happened. So there is a lot of oversight that ensures that, and everything in gene therapy is voluntary. Nobody forces you. Your treatment or standard of care will never change because you refuse to be in a trial. And then the patients have the right to just say walk away anytime,
Right.
anytime they can say, okay, I don't want to do it, and no questions asked. So they have that right to and when we give them the informed consent, they have, even after they have signed the informed consent, they've had them or their family ask all the questions, they've had time to review the informed consent, sign it, they're given a cool off period, and then we phone them and ask them, do they still want to do the study? They didn't make the decision in the heat of the moment, you know. And in the DSMB, the Data Safety Monitoring Board, actually reviews, or the medical monitor or the DSMB, an independent person reviews that the patient was actually eligible. We didn't just force somebody in that was not eligible for the trial. So there's a lot of safety features built in to prevent the horrible things that happened before from happening. Patients have all the rights to participate or decide not to participate in the study.
Thank you so much. So it's definitely a lot of regulatory oversight. But one of the things that when we were talking, and I had an opportunity to meet you, you said, and I wrote this down, there is no other way to improve therapy. We must learn and adapt protocols as things evolve. So why is it important that Black Americans get involved in clinical trials?
So as you've quoted me, there is really no other way to make progress. Sickle cell disease was the first genetic disorder. It was the first disease where the genetic basis was discovered over 100 years ago. And the only way we will make progress is where we have patients with sickle cell disease or patients with genetic diseases enroll in trials, innovative, potentially curative trials, so that we learn from that. We advance the field. We change the standard of care, which is inadequate, in my opinion. And it's time to change that. It's time to change the suffering of the patients with sickle cell disease. And the only way to do that is to do clinical trials and basic research. So I don't know if I have told you this before when we met, is that, when we were doing the proof of concept studies, it took us 15 years to get to this stage. So 15 years of research, we were laying one brick at a time in the wall. And we needed to get blood making stem cells from patients with sickle cell disease, because you can't fix a normal cell. We want to put in a gene, you have to see that you actually, in the test tube, you are able to prevent sickling. And so we had patients with sickle cell disease volunteer to give us, a small amount of bone marrow. And that's not trivial. I'm really grateful to all those patients, because undergoing a bone marrow for research, for the greater good of others, and those patients weren't going to benefit at all, from that particular donation. But they gave us a bone marrow aspirate so that we could go into the lab and figure out how to fix the disease. And those efforts have resulted in this gene therapy now reaching the patients. And then patients enrolling in this trial will then, further tell us what works and what doesn't, and how
Right.
to work well, and how to then make a new standard of care for this disease.
Thank you so much, Dr. Malik. Micaiah, I wanted to, just for the sake of time, just ask you, how were your questions and concern answered during the gene therapy clinical trial?
They were all answered. I was gonna say pretty well, but that's not what I was looking for. But they were all answered to like the best of the doctor's abilities in which they could answer any of the questions.
What kind of concerns did you have that they were able to answer?
Just like about who, like, when we were doing the final step of the trial, like, because we were from Columbus, like, is my mom going to stay at the hospital, or are y'all going to help us get a hotel, or is she going to Ronald McDonald, or like where, like questions like that. There was questions about scheduling because I didn't have a porch, so we had to get that placed, and a whole bunch of different other things. So any question that I had about anything, they answered. But like I said, I didn't personally have many questions, but when I did, they were answered.
And your program coordinator was available, you had their phone number, and you were able to get back to them. And they got back to you.
Yep.
Fantastic. So for the sake of time, I want to go back to Dr. Malik again, because I want to make sure that our audience has a chance to really have a chance, the opportunity to ask questions with you guys. What is important information does a patient need to know if they're interested in a gene therapy clinical trial? So what are some of the big picture steps if they're interested?
So I think that the easiest way, if you're interested, there are several groups doing gene therapy trials for sickle cell disease, and so you can go to clinicaltrials.gov. We had, I think, a clinicaltrials.gov. If you go to that website and you type gene therapy sickle cell, you will find all the studies that are going on and which ones are recruiting. And if you want more information about the trial that I discussed today, the one that I had designed, the approach, you can visit the website that's displayed on the screen. It's www.momentumtrials.com. Or you can, if you go to clinicaltrials.gov and you type in the number that's written, which is, every trial has its unique clinicaltrials.gov registration number. So if you type this number, you can directly go to this trial on clinicaltrials.gov. And if there are questions that you have, please post them in the Q&A and or get in touch with us via, you can get in touch with Dr. Anderson, and she will make sure that the questions reach either Micaiah or me or whoever else you want to, and you get the answers to those.
Most definitely. I will do that. So is there anything different about the trial or therapy that you are involved with?
Yes. So the other gene therapy trials that are going on, gene therapy and gene editing trials for sickle cell that are ongoing, they all use what we call the myeloablative chemotherapy approach, which is heavy duty chemotherapy that is given for transplant. So that wipes out all the blood making cells in the bone marrow, and then you give back the gene modified blood making cells or blood stem cells. The approach we are using is actually two different things about it. One of them is that we have modified the fetal hemoglobin gene to make it a super fetal hemoglobin gene, if you may. I mean, it really has a very potent activity in preventing RBCs from sickling. The second thing is we are using a reduced intensity chemotherapy because we did research in the laboratory where we found that you really don't need to correct every stem cell, every blood stem cell to correct the disease, because if you correct a fourth to a fifth of the bone marrow stem cells, the red blood cells that they produce are so much stronger, they far outlive the sickle red blood cells. And therefore, that's enough to cure sickle cell disease. So we think that, I mean, of course, I'm 100% biased, but because I developed this approach, but I think that giving less chemotherapy will have less side effects later. There'll be less issues with fertility, less issues with second malignancies that occur due to chemotherapy itself. Long term, this is 10, 20 years down the road. And less acute side effects, less hospitalization, less morbidity or complications associated with the transplant, less infections. So all of those things. That's why I feel that, I want this presentation to be unbiased, and we're presenting what we did because we did it and we are so excited about it. But there are many other gene therapy approaches that also work. They just use a different chemotherapy approach and different anti-sickling gene approach. So those are the differences.
Thank you so much, Dr. Malik. And again, if you go to www.clinicaltrials.gov, you'll be able to see across the United States, there are about 12 sites that are recruiting patients with sickle cell for gene therapy, clinical trials with different approaches, and also in France as well. So I encourage you all to visit the website to get more information. So as we get ready to kind of close up, I wanted to end back with Micaiah because your journey towards a cure has been just empowering for me, and I'm sure the viewing audience as well. But I wanted to ask you, what was the experience like being in a clinical trial overall?
It was most definitely different. I've talked to one of my friends because I've been trying to get him to do the trial since like I started, so it's been like a year. And I told him, I said, it most definitely was a different experience. Would I do it again if there was like a different outcome or like no outcome at all? No. Would I do it again knowing that I'm helping myself and other people? Yes. But like I said, it's most definitely, I told him it was most definitely different than anything I've ever experienced with sickle cell. So that's a lot to say.
Are you glad you did it?
Yes. Yeah.
And what were your biggest surprises?
The biggest surprises were, I feel like going into it, I didn't think about I don't want to say like the PTSD that's involved with sickle cell and like having stuff like that. So the biggest surprises was being stuck in a rainstorm and not ending up in a crisis after, or being able to be in a pool on vacation for a while. And I'm sitting here like, something's going to happen. It's going to happen. Like while I'm in the water and then nothing happens. So those are most definitely the biggest surprises.
Wow. So I want to thank both my excellent panelists here for a fantastic night of discussion about clinical trials for gene therapy. But in the end, I just wanted to have you guys say just a few words of advice to our listeners about gene therapy. So I'll start with you, Dr. Malik.
So I think gene therapy is the new way of treating genetic diseases, not just sickle cell disease. There are many diseases that are being approached. And time will tell if this therapy will replace bone marrow transplant or will be offered in parallel with bone marrow transplant. I definitely would urge patients to enroll in trials so that we move the field forward and we do good for our patients and develop novel therapies that can help them.
Thank you so much, Dr. Malik, for your time this evening. And now for our sickle cell warrior, Micaiah, what are your last advice or words to our listeners tonight?
I would most definitely say just do it. Go for it. There's nobody holding you back except for you. And there's nothing like with everything that, like, for the patients, with everything that you've been through with sickle cell, like, this isn't going to top it, and it's almost over. And like I said, nobody's holding you back but you. Do it for yourself, do it for the babies that don't have a voice yet, so make history.
Thank you. And I think we made history tonight.
You made history. Yeah. Thank you.
Yes.
Yeah.
I also
And
wanted
thank you
to
for inviting me. This has been really good. It's been so nice
Oh.
to hear from the other side. So I really, it's been enlightening.
I want to thank you, Dr. Malik, and I want to thank you again, Micaiah. You've been such an inspiration to us all. I also want to thank our partners this evening, the Sickle Cell Alliance Foundation, out of Columbus, Ohio, our CEO, Lisa Meacham, for providing me with resources, for informing me about the advocacy work that you're doing out in Columbus, Ohio, and also introducing me to Micaiah. It's been such a great opportunity and partnership for TOVA, as well as the Sickle Cell Disease Association of America. Thank you all for joining us tonight. So in closing, I want to say that the future looks bright for sickle cell. I want to tell you that having provided care for adults and children for 20 years, I've never been more excited for my patients to live the most optimal quality of life. And I think we are here. And I also want to thank Dr. Malik for persisting for 15 years to develop her patent and her approach to treating and curing these individuals with sickle cell. And I hope that as you continue to fine tune this approach, that it will be done in such a way to even have the opportunity for more participants to be involved and that they will be cured. We are in your corner, and we are excited about the future for sickle cell. So I wanted to make sure the viewing audience has the opportunity to ask questions. So if you can go to your Q&A, you can put your questions into the comments and we will field them to Micaiah, to Dr. Malik. And if there's something I can answer, I'll do that as well. I wanted to start with Micaiah. There was a question that came in from Mrs. Johnson. Are siblings always, there was another question about how often did you have to get apheresis after your bone marrow, after your stem cell? I'm sorry, your gene therapy trial.
After the gene therapy trial, I actually just went back to transfusions, and I had to get those for about 5 to 6 months after, but it was once a month or like maybe twice a month. And I actually have stopped those now, so I don't have to do those anymore.
So you had to get monthly transfusions for about 3 to 4 months after the gene therapy trial. Okay.
And that varies with the person though.
Okay. Okay. And then Dr. Malik, I'm fielding this question to you. Are siblings always compatible for bone marrow transplant? I've been told you have to have a full sibling, and there's a 25% chance that full sibling is a match.
Yes, you just answered the question too. So yes, 1 in 4 full siblings would be a match, and the sibling that is a match should not have sickle cell disease. Having sickle trait or having normal or normal is okay, but not as sick SS.
Thank you so much. And now Leslie Evans Johnson from Delaware has a question. Is there an age cutoff for gene therapy?
So the different trials may have different cutoff ages, but I think the trial that I spoke about that is being conducted by Arrowant has 45 years as the upper age limit. It's currently enrolling adults, so 18 to 45. And I think once they have had 6 adults enrolled and good results, then the FDA and all the regulatory agencies will look at the data and see if they can lower the age to children, and then children would enroll in the trial.
Another question from anonymous attendee said, will we be able to access the link and share have someone who really needs to see this? And the answer is yes. We will make sure that the viewing audience and the public has an opportunity to view this at a later date.
Before we go into the next question, I saw somebody said that it seems risky, and I just wanted to throw out there that life is risky. If you don't take a chance, like if you would have never ate French fries, how do you know you like French fries? Like, how do you know that you like potatoes, you know, or that you are allergic to something or have an allergy? Life is risky. You just got to take that jump, or else it would have been a shoulda, coulda, woulda situation, and that would be on your mind for like the rest of your life. So take that risk. I just wanted to say that. Sorry.
Thank you so much for sharing. I was going to get to that, but thank you, Micaiah. And I have another question for you, Micaiah, in the Q&A. How would you compare your quality of life after the transplant to before the transplant? And this is from Teresa Springer.
It's so much better than what it was before. I don't have any sickle cell related pain. You scared me. I'm sorry. My door. I didn't have any like sickle cell related pain, but I had like a lot of bone damage pain from the bone damage that I did have from the sickle cell, but quality of life is like a bajillion times better.
And I didn't get to ask this, tell the viewing audience, what medications are you taking right now?
None. Nothing.
None. Wow. Another question from Rachel Anderson, and I'll field this to you, Dr. Malik. Is the gene therapy approach covered by insurance?
No, not yet, but it's covered by the people doing the trial. So it shouldn't cost you anything.
You get paid.
Yeah, you get paid for participating in the trial for your time and effort, and you shouldn't be, your insurance is not charged. So insurance will pay only if, this becomes approved by the FDA as a standard of care or as a licensed therapy.
Thank you so much. And then I have another question in here, as someone that develops informed consent language for gene therapy, what suggestions do you have to make the language easier for the research participants to understand the potential risks? And does it have to be so long, these documents? How can they make them easier to read and understand?
So the length of the document has been debated forever, but there's nothing we can do to shorten it, because we are required to inform of every side effect, every pro and con. So there's nothing you can do to shorten it, but it definitely should be written in simple terms, so that and all the institutional review boards actually review the informed consent very carefully to make sure it is understandable by a lay audience, a patient. And so they are very, in fact, they review it very critically to make sure it truly reflects the honest truth, and it's written in simple terms. The other thing that at least our IRB required when we got this approved was that patients get the informed consent ahead of time so that they can read it, and reflect upon it. They can take it back with them. They don't have to sign it right away. They can ask questions. Even after they've signed it, they can come back and ask questions. And it's all completely voluntary. So those are the steps that can be taken. But I don't think they can shorten it, only because if you shorten it, you would have to delete information that's important for patients to know.
Thank you so much, Dr. Malik. And this is back to you. This is from Wilmer Rapanzo from the We Love You Foundation. Hola, ¿cómo está? He asks, what would you say to warriors that are currently considering undergoing this procedure?
So for the older warriors, like let's say like 30 and up, let's say do it. You've already experienced a lot of life, and this can add on to so much more with you being able to do things that you've never done before. And I'd say for my age, or like when I started the trial, I was 19, and they age you out at 21 at Cincinnati Children's. So for the kids that are like 18, 19, 20, do it before you age out. And do it before they send you to the adult hospitals, before they even get a chance to be able to discriminate against you in any way, shape, or form. Do it, so that you can once again live the best life that you can.
Thank you. Thank you. I think that we have answered all of the questions in the Q&A. If there's any more questions, please send them now, or you can just always comment on our Facebook page, hashtagging TOVA Community Health. And again, if there is any further questions that you would like me to answer, you can always reach me at contact@tovacommunityhealth.org. I think this it concludes our Surviving and Thriving After Gene Therapy, a clinical trial virtual forum. I am so excited about the future for sickle cell. And again, I want to thank Dr. Malik for the fantastic work that she's doing at Cincinnati Children's Hospital, and our sickle cell warrior, Micaiah, for sharing your journey. If you would like to contribute to TOVA Community Health, you can Cash App us at $TOVA Health. And I also want to let you know that we will be making a contribution to Micaiah's college fund. She doesn't need any medication. She has all of her medical care covered when I asked her, but she said she wanted some help with her books. So we want to make sure Micaiah graduates and continues to live the best quality of life she can. And remember, she still has to go back every month to Cincinnati for monthly visits and surveillance. And this may go on for several years. So we want to make sure that she's able to do that and has transportation to do that as well. Again, I thank you all for coming and joining us this evening. This has been a great forum, and I look forward to trying to provide more information about gene therapy and other disease-modifying therapies so that we can offer these options to all of our clients and patients with sickle cell. You guys have a great evening. God bless.